I am just so pleased to introduce to you Dr. Norma Alcantar. Dr. Alcantar is a National Academy of Inventors Fellow, and she comes to us with many years of experience and funding, federal funding. When she was inducted into the Inventors Hall of Fame, the selection committee just lauded her pioneering work in engineering of natural products to create sustainable and non-toxic technologies for a wide range of applications. Dr. Alcantar holds 22 patents, 12 of which are related to the cactus fuselage that she uses as a purification system. And this was a folk practice that she learned from her grandmother in Mexico to boil the pads of the prickly pear cactus and to produce drinking water, clean drinking water. Professor Alcantar comes to us with chemical engineering and biological materials experience, and she's been here since 2003. We're very proud to have her present today, and I love the advocacy she has been doing for research, and includes mentoring students in her research lab, and students have actually progressed from her research lab into the research park with small businesses. So without further ado, I am very pleased to introduce to you Dr. Norma Alcantar.
Well, thank you, Sandy, for that introduction. And thank you all for being here and listening to what I had to say about really just my experience with some of the funding agencies, especially NSF. My background is some basic science, how to relate intermolecular forces in material science to practical applications and structure, and how the relationships between structure and property can give us insightful information and innovation. So I'm pleased to present my work that has been recognized by NSF, and then I'll give you a little bit of my experience with this topic and, just in general, my journey through academia. So let me start by saying this is kind of like, it gives you an overview of my research is on functional sustainable solutions for water purification systems. We start with the Opuntia ficus indica, which is known as the prickly pear cactus, or nopal in Spanish. From that, we actually extract, let me see if I can get it. We extract what is called the cactus mucilage. This is a substance that when you open a cactus pad, you will see like a viscous liquid, and that can actually be extracted. We've seen that it has, depending on the extraction method and the composition, has different kind of nanostructures. We have been able to play with these materials to make fibers. These fibers can actually be made into membranes, and the membranes can actually be put into a filtration, which is called point of use filtration device. So this is kind of like, can you see that? Oh, sorry. I need to change that. Okay, so let me just show you a brief video. The National Science Foundation prepared this video to really showcase my research in 2014, I believe. So let me show it to you. Can you hear the volume?
The prickly pear cactus salad. It's a delicacy in Mexico, but it seems these cacti have uses beyond the culinary, cleaning up oil spills and purifying water.
You can see how It's really easy to grow.
With support from the National Science Foundation, University of South Florida chemical engineer Norma Alcantar is using the gooey flesh of the cactus called mucilage to clean up polluted water. Her interest in green chemistry came from her grandmother, who told her of using cacti to clean river water in rural Mexico as a young woman.
They would take the cactus plant, but they would cut it, they would boil it, and they would use the water from that preparation to clean the water.
When Alcantar tested that process in her lab, she was amazed at the results.
I couldn't believe it. I was like, are you serious? It really works? Because I thought I will see an effect, but I didn't think I will see an effect so dramatic.
These biobeads are made of mucilage.
This is the controlled initial concentration of the methyl blue dye versus the one that has been in contact with the beads.
After an oil spill, cactus goo could be an alternative to chemical dispersants like those used in the Deepwater Horizon cleanup.
The same action can be done with a very low concentration that makes it very sustainable and fairly inexpensive.
Alcantar did water quality tests in Haitian refugee camps after the 2010 earthquake, finding high levels of heavy metals and bacteria in the drinking water. While there, she kept an eye out for cacti, and they were everywhere.
This is another gas station.
Tests back at her lab confirmed that mucilage could successfully clean the polluted water from Haiti, and that's significant. In the future, cacti could be harvested locally to decontaminate drinking water quickly after disaster strikes. Using a natural product to clean water, it's a sharp idea. After all, there's no reason to get stuck on the old way of doing things. For Science Nation, I'm Miles O'Brien.
So you can see that this kind of material can be used not just to separate sediments, but also we have used it to separate bacteria. It forms these flux that then are easily removed with any kind of filtration device. Sediments, as it is mentioned in the video. We also tested with heavy metals. So this is arsenic iron solution. And within 10 minutes, you can see that it actually separated the iron to the bottom, to the iron and the arsenic to the bottom of the container. And we also used it in 2010 as part of a base for a natural dispersant that could be used instead of commercialized dispersants that had a negative effect on the health of marine organisms. So let me, so how my research had related to NSF programs. This is how I wanted to connect it. So essentially there are different divisions. So the work on the separation has been funded by process engineering. Then we have reaction systems. There's another division of NSF that we can send it to. And then also molecular thermodynamics. So a little bit more on the basics of how materials work. This is part of the CMMA, and the other two are on CBET. So let me give you a little bit of an introduction on the mucilage and how this relates to awards from NSF and different patents. So the Opuntia ficus indica is the material we use because it reproduces really fast. So we want to have a material that can be sustainable. These are the different awards that have actually been provided by the National Science Foundation on the different parts of my research related to cactus and the separation of contaminants. So you can see each one of them actually lead to another one and another one and another one. They kind of like build up throughout the years. And with that, also each one of them actually produce a patent. So some of them have been double, like this one. And you can see that your research had to relate to part of innovation. That's always what I try to teach my students. Try always to see what is the IP and how innovation is related to the research and the application. So thank you to NSF for all this money. Also, I wanted to let you see, why this is important, why it has a material. Well, it's a thick gum-like substance. You can see it out here. This is part of the extraction process. I have showed you that it precipitates heavy metals, bacteria particles. This is because it has specific surface active characteristics, and that's part of the phenomenon that we study and how can that be applied. And then if you also see the cactus is a substance eatable. So there's this is the part of the cactus in the plant where it stores water. So we take advantage of how the biomimicry, which is the science to study bio-inspired materials, how those relate to making an invention. So part of the extraction process that we use, we macerate the cactus pads into either the solids, and then we separate the liquids from that. From the solids, we obtain what is called the gelling extract, which is one of the fractions. And then from the liquids, we obtain what is called the non-gelling extract. And then, of course, you can have the combined. So let me tell you a little bit of how we started this research. So initially, we started looking at how can we separate sediments? Why this material will be better to what is existing right now in a water treatment plant. So one of the studies we have is the flocculation rate. So that part of the research where you separate contaminants from water. So we have a column that had dirty water. This is a concentrated solution of kaolin. Kaolin is a ceramic particles. They are about 500 nanometers in size, and concentrations are really, really high, right? 5 grams per liter is a lot. So this is more like mud. Then we actually add the flocculant and the cactus mucilage, and then we look at how the column, the liquid solid interface between the water and the dirty slurry can separate with time. And then we can actually plot that and obtain data that will tell us how this is better with respect to what is existing right now. So let me tell you, this is the fall in interface height measured by centimeters, and depending on how long does it take. So the first line, the line that you see out here, is the control. So nothing has been added. This is just leaving it together. And you can see that it takes about 20 minutes to separate 10 centimeters. Now, we try aluminum sulfide. This is one of the commercial flocculants that is used in the water purification system. It's much better. So for the same amount of separation, right, it takes about 16 minutes. Now, I'm going to show you the one with the GE. Wow, right? So to the same separation, 10 minutes, 10 centimeters, it actually just takes 6 minutes. So can you imagine the process of water separating contaminants for water is much, much faster. If you use the one that we also call the combined, that would be in between the gelling and the non-gelling extract that I mentioned before. So this was very exciting, and we discovered that all the extras were much faster than aluminum sulfide. Now, how do we measure this? Well, because you can actually look at the specific slope, and then you can transform that in a rate, right? So that would be centimeters per minute for each one of the components that you are trying to study. So we found that the GE is much, much faster than aluminum sulfide. The NE is still much faster, and the combined is still much faster. But what is it that we call faster, right? So now here you can actually compare apples to apples, and we found that it was 3.3 times faster than what is used commercially. Now, that's not important also in terms of how much do you use, because to obtain the same kind of rate, selling rate from aluminum sulfide and GE, guess what? We discovered that you will have to use about 3 grams per liter of aluminum sulfide. Well, we actually are using 0.01 milligrams per liter of the gelling extract. What does that mean? Well, that means that we are using much, much less of this flocculant. We only use a dose that is about 300 times smaller than what is needed or what is actually compared to what a flocculant from industry will be. So why is that? Well, for NSF, it's very important that you understand the background and the basic information. So we discover the structures. You can see the gelling extract has more like a lace-like structure. The non-gelling is more like a fishing net structure. This is because it's produced by different polysaccharides. It's a combination of polysaccharides. In the case of the GE, you will have, we actually were able to determine that it has about 56% of what is called uronic acid, which is one of the high hierarchy sugars, where the non-gelling only contains 11. But the gelling extra has more arabinose, which it has the non-gelling has more arabinose, and the gelling extra has less. So another part of why this was very attractive to NSF is because we could present the data in a way that they could see it easily. So this is the work that we did for using the mucilage as a dispersant of crude oil. This is the control, so you can see it's just a blob of water and oil in the water. When you add Corexit, you form these particles, and these are the particles that are biodegraded by bacteria. When you add the mucilage, then you see the particles are much smaller. Smaller particles then relates to higher stability, and that you want that in a dispersant. And then we try different formulations with a solvent that is a green solvent and green particles, and then all together. And we can see that if you have used the mucilage or the mucilage with particles and solvent, the stability is much, much better. So we were able to go through a process where you understand the different properties of the material. This is so that you can see it easily, right? You in a dispersant, you want the solution to be all throughout the column of the water so that it can be available to bacteria for degradation. The control usually it just separates it on the top. If you look at Corexit and mucilage, they actually go head to head in most of the experiments, even if you shake it just for 5 minutes, 3 hours, or 6 minutes. Okay, so all of these you can see as part of my research. This is just like a tiny little slide throughout time. I can tell you that we have learned the composition of the mucilage, which was unknown. We also know now that what is called the lethal concentration, 50, which is part of how you measure toxicity for NE is minimal, versus Corexit, which is much, much higher. And we measure this with some marine organisms that are usually a benchmark for these kind of experiments. So anyway, this is part of this research, and it has been funded by the National Science Foundation. But really, I want to leave you with a message of, throughout my career, what is it that I have learned? I wish I have known then, when I started in 2003, what I know now. And some of these are mentioned here. So it's very important that you who to trust in your same department, in your same division, that you recognize who's willing to be a mentor. You can have a mentor for, if you want to go after federal money, you can have a mentor to mentor students. You can have a mentor on how to deal with life issues. So you can have many, many mentors. So recognize who's actually willing to give you advice and good advice. How to choose my grad students. Grad students are very important. I wouldn't have been able to accomplish all the things I did without the help of my grad students. So how to choose grad students that are dedicated, that persevere on what they had to do, and that are good scientists. That's very, very hard. How to efficiently prepare my classes, because you had to do research, teaching, service, all of that. So how to spend an effective amount of time in all three. Related to teaching, how to improve student evaluations, how to actually provide the students with the learning that they are expecting. How to pick postdoctoral fellows. That's very important, right? Because the postdoctoral fellow is the one that's going to help you in your lab, in your experiments, in your research to get ahead. But also collaborators, how to go outside your department and get collaborations, how to select who do you want to work with, and how to protect your ideas, right? Did you come up with the idea, or was it an idea related to the collaboration? How to protect that. So also, I wish I have known then how to approach program directors. You had to build a relationship with them. You had to start talking to them, send them the summaries that you prepare for your grants. Talk to them. They are there for you. They will be able to talk to you at all times. How to participate on panel reviews. You will see that who is in the other side, who is actually reviewing your proposal. How to name out different reviewers for your papers, right? This is how you want to have a paper published. Who is going to be reviewing your paper? Well, it has to be someone who's really giving you the advice to produce a good document at the end. And also, it's not just giving you a review that is for the heck of the review, right? You want something that you will be able to modify and change that to the best of your abilities to get your research published. We all have been in situations where you have unproductive members, so how to get rid of them, how to actually be effective at providing resources for that so that they can leave your group, and also give you the peace of mind that you deserve, because you don't want anyone unproductive in your group. Sometimes, and this is something I have been, this has been some of my experience, sometimes the staff personnel is rude, or they actually are having a bad day. So how to handle that? You had to be very polite, you had to be very diplomatic. So how to learn to deal with situations where everything can be unexpected. Also approach your boss, right? Your chair, your dean, your advisor. And all of these, being in academia is very stressful. So how you can release the stress and how to pick your battles. So some things that I did that I shouldn't have done, I recognize that now, is that I wrote a proposal for a center in my first year. That was a big enterprise, and I probably shouldn't have done it. Usually they want material centers to be led by experienced researchers. So I got very good reviews, but most of the critique in the proposal was that I was just an assistant professor. Something that I shouldn't have done is to also organize an international conference. It was good. It was a lot of work, but I shouldn't have done it. But this is in your first 5 years of your 10-year track. You should concentrate on your research, teaching, and service. Organizing an international conference comes next, right? I also collaborated with senior faculty that took advantage of my resources and opportunities. I know now how to deal with that. I didn't know at the time, and I felt that I didn't have the power to say anything. So it was not very effective. How to listen to your gut. If you feel that your gut is telling you not to do it, or do it this other way, or go and talk to that person, listen to, to your internal self. I also let the apathy from some of my colleagues to get me down, and that should be, right? You learn that through the process. Things that I did right, I think there are more than, but I shouldn't have done. But I actually asked for what I needed. I got a huge lab because I asked for it. I approached good collaborators and sought good mentors. That was very important. I looked and started a simple project, which is the one for the cactus, that actually gave me lots of products in my first 2 years. Something that was easy and that I was able to publish right away and learn and get money, federal money, right away. So I started to compile my 10-year package in my first year. Do that. Don't let it wait until you actually have to do it. Start compiling everything of your package in your first year. Have a file, have a directory, whatever you want to call it, where you start organizing all the things you do. And then I was determined to help and motivate underrepresented female students. That's something very close to my heart. And that's something that I took as part of my responsibilities. I always participated in institutional programs to find out what those are in your institution and get on board. I took advantage of any kind of seed money, institutional opportunities. I also was instrumental. I grew up in Mexico, so I was able to start an agreement with Mexico universities. So I did things that I actually know how to do it to benefit my career. This is part of that last point. I did not wait to have a life and family until after tenure. I have my son on my 4th year, and that was very important to me. So I simplified my life by actually having a crib in my office and took advantage of any kind of external commodity that you can think of. So my recommendation to you is, if you make a mistake, well, everybody makes mistakes, just recognize them, fix them, and then move on. Don't let them drag you. Sometimes you need to make a hard decision, take the responsibility and courage to do so. And this is very important. If you get upset, because there's going to be times where you get upset, write an email to yourself where you write everything that you're upset about it and send it to you. Don't send it to anyone else, okay? Wait for some time before you take action until you come back. This is the best advice I got from one of my mentors. And so I hope you have a journey, a wonderful journey in your tenure process. These are part of my students throughout the years. As I mentioned before, they are really the ones who did all the work and deserve all the credit. These are some of the supports. You actually saw all of them before. These are the ones that I have seen in the last 3 years, and also the Gulf of Mexico Research Initiative grant. So with that, I will take any questions and let me know.
Thank you, Dr. Alcantar. That was wonderful. David is going to moderate the question and answers, which are, the questions are posted in chat. So, David?
So Dr. Alcantar, how did you modify your proposal framing as your work went from launching to maintaining your research pipeline to continue to get grants over many years? How did you keep your subsequent grant proposals innovative, as you titled in your talk?
Well, the more you learn, you start with a small project, as I mentioned before, and then you're going to be learning different things and discovering different phenomena. And then from that, you're also going to be able to pick of what is important from your discovery and then take that information into a different system and then explore that. That worked for me. How to be able to recognize some discoveries and how come then we were able to take it to the different other applications that resulted from the research that we were doing. I listened to my students. One of my first mistakes I did, I'm going to tell you. One of my students was doing research on Alzheimer's, and she said, we should try the mucilage to see if it works for Alzheimer's. And I told her, no, don't waste your time. It's not gonna work. Let's just concentrate on your project. And 10 years later, we try it, and guess what? Because the mucilage has the ability to separate large hydrocarbons, it also has the ability to separate the amyloid beta fibers that are formed in Alzheimer's plaques. And I didn't know that at the time. And I wasted 10 years because if I would have listened to my student, we would have started that much, much earlier. So I'm telling you, listen to your students and take them, give them the credit they deserve. Most of my patents are not alone. Always, if I have a patent, I will have a student related to that patent, and that's because they are also discovering, right? It's part of their graduate education. They're doing discoveries every day, and they are the ones who will be able to give you the details. They know exactly what is happening in the system that is related to the structure or related to the property that you are studying. So give always credit to your students. That's always very important.
Thank you very much. And someone wanted to thank you for the information on the prickly pear cactus. They're wondering, can you tell us whether the spineless variety is also useful for water filtration in the same way?
Which variety?
Spineless.
Spineless variety.
Okay, so, well, most of the opuntia is the one that works for experiments. But you can also take okra, and okra has mucilage, and it will work similarly to what the prickly pear is. So you can also do that as well. As long as you are utilizing this gooey part of the cactus, this is as I said before, is the substance that stores the water, right? So any cactus, or any cacti can actually survive in the desert because it has the ability to store water effectively. So that part of the cactus is the one that can help you for water purification.
That's perfect.
Terrific. Now, you talked about protecting your ideas, and the issue seems to apply, there seems to be no way to protect those ideas without disclosure as a young scientist. So what type of advice might you have for a young scientist who feels the need to disclose in order to apply? Well,
Find out what the procedure is at your university. And then meet with the patents and license senior personnel and explain your idea. Everything that you say to them is confidential. They do a preset or pre-search on if your idea is going to be important or not. And then they will come back and say, yes, this is something we can patent, this is not. Now, always do that before you send a manuscript for review or before you send a proposal. Make sure that you know if the idea that you're sending can be protected. You have a year before you disclose that information to actually file for an application or disclosure. So you have a whole year. But if after the 12 months, if you want to go back and disclose something, you will not be able to do it. So time is of the essence to protect your ideas. So make sure you are familiar with what the process is. It's complicated. It takes time to learn, but that's something that you need to do.
Great. And how hard was it to apply to NSF? I mean, are there any recommendations about applying to the sponsor?
Well, as I say, build up a relationship with the program officer. It is really hard. I mean, I'm telling you, funding rates at NSF right now are less than 10%. That means that you have to write about 10 proposals and one of them can be picked, right? Those are the odds. So in a year, writing 10 proposals is a lot of work, right? So make sure you have the right kind of information. I show you a little bit of how to present it to the reviewers so that they can be actually wow, like, oh, this is so important because you only need tiny little amounts. This is something that we didn't have before, right? So you have to be able to express that in the proposal. You have to wow the reviewers. They have to actually see the research you are presenting that is totally novel, that nobody has been able to do it, and that you are the perfect person to perform that research.
Great. And so now, at what year after being an assistant professor did you start your lab? Did you wait for a federal fund first, or did you just start?
I started my lab when I arrived to USF, and I'm telling you, it wasn't ready. I only had one sink to work with, so it took a couple months. I would say about 4 or 5 months to have a lab actually ready, and it was working, every day. If you're in academia, you're not going to be able to work 40 hours a week. I mean, I don't remember in my life now when I only work 40 hours a week. I think my days or my weeks are 60 to 70 hours a week. So it's hard work, but I started right away.
And how did you benefit from publications to alter your proposals to the NSF?
Well, so as a student, make sure you have publications, right? And that gives you validation that you are capable of doing research. Once that you start a proposal, make sure that the discoveries that you have, that you publish the work. And it's hard. It is hard because you want the students to do it, but the students don't know how to do it, right? They haven't done it before. So you have to train the students to publish, and you have to get the data to put in the paper. So I learned the hard work. At the beginning, I was writing the papers for them. And then one of my students said, no, I want to write the paper. I just don't know how. And it takes more time. But once you teach one of them how to do it, guess what? They actually then teach the next student how to do it. And then they actually create a environment in your lab where now everybody is competing against each other to publish more. And that's what you want, right? That you actually want to build up peer pressure, healthy peer pressure, so that can be attained.
And do you currently have opportunities for students to join your research efforts?
I do. It depends on the skills and the level of their where they are in their career. If they are ready to be in, has a master or has a PhD, or I also hire, as I mentioned, our REU students, research experience for undergrads candidates. And that's because I want undergrads to be able to learn how to take data, how to be able to modify a process, how to be able to think, what is next on the steps when you are making an experiment or separating something, or what is it that you have to use, right? You have to use your hands. What is the best way to do it?
And do you have a specific process, do you have guidelines that you use for sending your summaries to program directors?
I use the same guideline that NSF has, what is the intellectual merit? Follow that. They have different questions that you have to answer. So make sure that first you write the questions and then you see how your research applied to those questions. And then from there, you change and modify the narrative to make it seem like a summary. And that's, present what is the novelty? What is it that your idea is doing that will modify existing knowledge and that will create new knowledge? What is it that you're doing that nobody has been able to do? What is the differentiator that your research is presenting? And how that is going to be possible with the methodology that you are suggesting. So all of these questions had to be answered, and it's hard. It's, you have, to me it works. You can see, I write everything, all my thoughts. I always writing or doodling on my thoughts. That's something that I just learned, because sometimes you are having lunch and then you think about something and you don't have anything to write. So, I can tell you, I have like different little post-its, right? All over my desk where I just write things that just come up to my mind on how to present the research. So that works for me. That may not work for you, but what is the method that works for you? Maybe your method, I have a colleague that used to use post-its, and she had post-it notes all over. I don't personally like post-it notes because I lose most of them, but that's what it worked for her. So, what is it that works for you?
What could you do if your department doesn't have research students?
Well, if I will invite undergrads to work with me. I think there are lots of students that want to learn, and they just want to learn something. Start with something simple, right? Just one question. What is, why is this material behaving the way it's doing it? Do we know that answer? And if you find that the answer is not well understood, or there's like many different ways to explain it, then you have something, right? You can actually pick a line from there and start something new.
And we'll take one more question, Dr. Alcantar. There's some in the chat yet, but we'll address those with you later. Do you have a specific application on your mucilage that has been applied to solve a problem in the Tampa Bay area? Are there any local applications?
Well, I don't know if many of you know, I actually started a company. It's called Water Health and Sustainability. And that's because within Florida, there's a lot of producers of fish, right? Fish farms. And one of the work that I started doing in 2012 relates to water purification on aquaculture. So that's something that has been applied in Tampa. I had a project with Marine. We created pilot units to separate. You see, when you farm fish, because the way you are doing it, you have fish on tanks, they start attracting molecules that are produced because in the tank you have waste and you have food, and that produces bacteria. Bacteria then produces different compounds that are absorbed in the skin of the fish. And that's what it gives the fish smell, right? Like when you eat like trout or any kind of fish that has been farmed, you kind of smell it, right? It smells fishy. So we have been able to have a system that can get rid of these compounds, take them off the skin of the fish and clean up the smell. So that enhances the quality and the production, so that we have been able to use that here in Tampa.